Listo.
Okay, today I will talk about a topic, so I will present a slide. You can see my screen.
Yes.
Yes.
Okay. The topic is transcription factors, orchestrated dynamics interplay between genome topology and gene regulation during the cell reprogramming. It's a large title. To understand all these topic, is important to know some definitions. The first is that genome, the organism's completed of DNA, including um, its genes. Also, the somatic cells are any cells in the body that are not gametes, sperm, or egg. Germ cells and cells going to become gametes or stem cells. Also, it's important to know the pluripotent stem cells because they have similars with stem embryo cells that allows generate or repair damaged tissues. No? The main purpose of this article or this experiment is reprogramming somatic cells into pluripotent stem cells by transcription factors. So practically, this will convert into these pluripotent stem cells. The article doesn't say why they did this, but the purpose of this is reprogramming somatic cells. The topology organization of the genome is divided into the levels. The level 1, that is divided into compartments. The A compartment, which corresponds to active chromatin, and the B compartment is the inactive chromatin. Then the second level of the topological organization in the genome is consists of topological associated domains that are TATs and chromatin loops, but this article is also focused in the TATs. Transcription factors is a way that they will turn these cells. So the abbreviation of these transcription factors that they will use is OSKM, and this contains NANOG and SOX transcription factors. The experiment consists in they exposed a mouse bond of the provided pre-B cells. So in this image, we can see the mouse, and these are the transcription factors that they will use to convert the cells. The B cells are the somatic cell, and this is the method CEC to generate the B beta cells as this, the orange. And then they activate the transcription factors to induce the programming of this, and this will in 6 steps. This, and these are the transcription factors. And the PSC is the somatic, no, the pluripotent stem cell. So this is like the process in general that they did. This can cause a change in the expression of the cells. So this is a somatic cells, the levels of all the steps. And there are like high in the blue one, and this is low the expression, and in the pluripotent they increase. So is important this. Also, as well as I said before, the second level is divided in compartments. So the A and the B compartment, and this is important because in the pre-programming they will switch. So this is like the B compartment and the A compartment. They switch is frequently in the reprogramming of the cells. So the 20% of the genome changing in the compartment at any time point during the reprogramming. As I said before, the chromatin state and the gene expression changed. So this doesn't explain very well why this changed, but it's like actually that this will change. Other important things is that the genome has boards in the cells. So these boards are important because they contain a large number of genes that will cell typical specific functions. So a cell can be used for immune system or development biology. The words is detecting by a lot of times in the reprogramming. Another thing that are important is the connectivity between these cells because the tats will in contact. So this is another important thing. The discussion that they have is that the dynamics genome topology, the chromatin state, and the gene expression is closely linked, as we said in the other slides. This linkage occurs in non-manager and change in the subnuclear compartments and the connectivity and that word. The genome of the topology, as in an instructive force that facilitated transcriptional changes, may represent a general principle and cell decision. So basically, is that I don't know if anyone have questions. No. It's a difficult topic because have a lot of genes and a lot of things. But it's interesting.
Yeah, so I want to ask you something.
Uh-huh.
In one slide of your presentation, you showed us that for the experiment, bone marrow from mice was used, right?
Right.
So can the experiment be repeated in any other tissue that contains stem cells?
I think that the stem cells is basically take to these bones marrow thing. So I think that in other parts of the body it doesn't have these stem cells. I think other, but I think they use this.
Okay. Thank you.
Yes. But I think that we continue with the other if you don't have any questions.
Okay, let me share my presentation. Okay, can you see it? Okay.
Yes.
Yes.
So the article that I read talks about the evolution, well, the influence that of protein diversity in the family of Oryza, which is a plant, due to the novel non-coding regions of DNA becoming protein coding. So yeah, basically what the article talks about is that there is the possibility that has existed through millions of years, not only in Oryza, but many other species, where there can be the generation of new protein coding genes that arise like completely out of nowhere almost. That's almost what the previous scientists thought, because they come from non-coding DNA sequences from their ancestors. So this, as we can imagine, leads to an increase in protein diversity because, well, if there was a region of DNA which could not be coded for a protein, and then the next species diverges and is now able to use it as a coding region of DNA to make new proteins, this might be the original reason why these two species diverged and therefore speciation occurred. So it is an interesting theory that has not been able to be proven correctly throughout the years, because scientists were skeptical about the possibility of this happening through evolution, and this would really change the way we see evolution as a whole. And well, the scientists of this article specifically had to look for high-quality genomes of 13 closely related Oryza species. As we can see in the image, there is like a very wide variety of Oryza species, and therefore each one of them have different qualities in their genome that made them diverge from each other at different points in history. And however, this scientist has had to research in order to do this this study correctly. They had to go and look for high quality genomes. What does this mean? That they had to take those genomes that were mostly done, like they had no gaps in between the chromosomes where they didn't know what that part of DNA coded for, or errors, or different things that nowadays not all genomes are complete. Not even the human genome is perfect, because there are some different variations from one like platform to another. They may have these non-coding regions different variations, and well, they had to diverge recently so that they could make an accurate timeline of how these genes apparently just appeared out of nowhere into their protein coding genes. And so the difficulties that this research faced were the fact that, well, it has been difficult throughout the years to validate the novel origination of proteins. And well, this is because if you think about evolution, there had to be units of evolution, which means that there are certain genes that had carried on from one species to the next because they made it the fittest. And if you have a gene that does not code for anything, then this is not really a unit of evolution, as it did not make a difference for this species to have it or not. So it was very weird to think about this, the novel origination of proteins, to be part of an evolutionary system. Therefore, there was the term was generated for orphan genes, which means, as we can see in this image, that instead of having an orphan genes from duplication divergence, which means that there was a species, and then the species had certain divergence from their strands, and therefore there was one that kept the same genes, and then there was another one with modifications, and then because of mutations, these small modifications in the next generation became total, which made it to look as if they had complete divergence and no longer any homology detectable, and therefore that's why the blue and the yellow are completely different. However, in this case, what really is supposed to be happening is that you have a non-coding gene, which is why you don't have like this line here, which goes for translation and transcription. You don't have that, but then all of a sudden there is a certain mutation that makes this gene a coding one. However, this has an open frame, but as we can see, it is not an arrow, which means it does not code for an actual protein. And then because it has certain parts of the gene that could be coded for something, later on there's another mutation that allows it to be translated into an actual protein eventually. And so this is very random, and not many scientists stand by this theory because it is not very supported. However, the scientists in this article managed to make it more reasonable. And so what was really needed in vitro to do this experiment is that, well, as we said, they had the high quality reference genomes, which were 13 from the family of Oryza. And then they had to understand the possibilities of turnover mechanisms, which are what retards, maintains, or accelerates the rates of DNA differentiation between populations. So basically, what are the factors that could have made the populations diverge over time? And also, there needs to be evidence for transcription and translation to happen, indicating that the new genes can be functional and produce not only the mRNA, but eventually a protein. So this was all made through this study via different platforms in the internet. They from a wide variety of platforms that contain the Oryza genome, they only decided to base their own in this Oryza map alignment project, which is this one. And this is because they realized that they had the least errors and gaps in their genome, and that's why they thought it would give them a lower range of error when actually coming to conclusions regarding the homologous genes of these 13 species, which are these ones on the right. And all these species diverged within the most recent years, which are 12.1 million years, except for this outgroup, which is Lersia perrieri, which is the oldest of these variant species that exist nowadays, but it is supposed to be the one that diverged the longest ago, which is 14.3 million years ago. And so this is the outline that they did in order to understand how these genes managed to diverge through time. So they thought that at first they had an ortholog non-coding region of DNA, which happened to eventually become an open reading frame. So as you know, when there is a non-coding region of DNA, this is not usually an open reading frame because it does not have to go through translation or transcription. And this is what first happens, that the orthologous becomes an open reading frame. And then you get another variant. And then they lose this indel. That is what made it, this is a section of DNA that made it not possible for transcription to occur because of the machinery that has to go beyond the start codon. This indel does not allow it and had a sudden stop in the transcription. but then it is moved away and then transcription can occur. However, this does not generate an actual protein. So this is why this is not where transcription really is taken into account in the history of Orisa. And then eventually there is an origin of transcription, and then there's the origin of coding availability, which made it possible for the actual species that nowadays have these functional genes to have them and have these new proteins de novo. The focal species of this de novo gene is actually mentioned to be the Orisa sativa subspecies of indica. And well, it is very interesting how it can be shown in the genome that there was a certain evolutionary process by which they started losing certain sequences that allowed this gene to, by almost a casualty, to have the actual de novo gene. And well, these are some of the examples of how they managed to do it per gene. This is down here is the specific gene they were studying, and these are the different species. As we can see here, the actual origin of transcription does not happen until the last subgroup of this species, which is the O. sativa japonica. And however, if you compare this gene to this one, the origin of transcription happens all the way since the first 14.3 million years ago with the first species that diverged. So we can see that each gene has a different process by which it diverged. And well, what they accomplished with this study is to actually understand what gene expression affected these de novo gene sequences, and what mostly affected in structurally speaking was the anther of the plant, which is the part of the reproductive system of most plants. And out of 57 genes, 24 were only for the anther, 4 were only for pistil, and 0 for the leaf. This means that there was a certain pattern for the novo genes to code for anther proteins, which is also interesting. And the scientists do not know how to explain this, but they do state that it is interesting, and further research should actually try and understand why this happens. And well, the results is that they found amongst the complete genome at least 175 de novo opening frames in the subspecies of Japonica. Another important result is that through mass spectrometry-based targeted proteomics and ribosomal profiling, they found evidence for translation, which means that these de novo genes actually work, of about 57% of the whole de novo genes found. So which means that 57% of all of these genes actually work into making proteins in the plants nowadays. And well, around 51.5 de novo genes per million years were maintained throughout history, all the way up until 2018. So it is interesting how this really is an evolutionary process, because it has somehow retained in the Orisa genome throughout millions of years. And well, in conclusion, this study actually provides important information to prove how de novo origination of proteins is responsible for protein diversity amongst different families of plants or other species. And this also provides evidence for of ancestral origination and translation, and how diversion of a species occurred into making the most recent subspecies of this plant. And yeah, that is everything. I don't know if you have any questions.
In one of your slides, you showed a cladogram, right?
I think it's more than one. That one?
Well, you showed one in which you showed like the 13 species of plants you were talking about, right?
Yeah, these three do. Why?
So this novo thing would be like it's the appearance of a gene without apparent ancestry, right? Or without.
Yeah, that's how it would appear, but really it's just like the ancestor had a gene that did not code for anything, and now the plant that lives today shows this same gene, but now it is functional and it produces proteins, and it is part of their what made them diverge from their ancestor. That's pretty much it.
Okay, so then these plants are still very similar in their genomes, right?
Yeah.
Okay. That was my question.
Yeah, that's what's interesting because their genomes are really, really similar, but what makes them different is the function of these specific gene sequences.
Okay.
And are the like different species located in different like places in the world, like have like different environments or things like that make it like adapt to that specific environment, or is it just like a random mutation that occurs?
No, it is not random. For example, as you can see, the O. sativa subspecies indica compared to the O. sativa subspecies japonica, they are almost the same species. However, they are present in different parts of the world, one in India and one in Japan. So they have different, both of them now code this de novo proteins, but they have different proteins. And I think it does have to do with the environment that they grew on, because these specific genes help them adapt better at their specific parts in the world, I think, which influenced how they evolved and become a different species from each other.
Yeah. And do you think there's like another organism or another, yeah, like another organism that uses this like mechanism for evolving, or that shows in general?
Well, in the introduction of the article, it actually talked a little bit about it. And it did say that there were many species that have ancestry in common, like in mammals. I don't recall which was the actual species or family, but it was a mammal, and they said that certain proteins coding for, I don't remember what, but actually had this same type of situation, and there was no like scientific research proving that this was what was happening. But the scientists in my article actually like suggest that this might be the reason for their divergence as species. So yeah, it's probable that others have the same, like, I don't know.
Like, yeah, the same like evolution mechanism. Yeah.
Mm-hmm.
Okay.
Yeah.
I think there are no more questions.
Question? Okay, then I'm just gonna stop recording.
Mhm.
Where do I stop recording? Huh.
Arribita. ¿No te sale?
Uh,